And here is the first session. The speaker, Professor Ravikant Upadhyay, is not coming. In his place, Dr. Mukesh Sharma from Ajmer will speak. His topic is non-homologous enjoyment.
A very good morning to you all. Ladies and gentlemen, this is the third day and the first session. And for this, we have Dr. Mukesh Kumar Sharma. He's from Department of Zoology, SPC, Government College, Ajmer. And the topic which he will discuss is non-homologous end joining repair pathway of DNA double-strand breaks and its application in cancer radiotherapy. For this session, we have chairpersons, Dr. Hemant Parekh, who is also the organizing secretary, UGC Research Awardee, Department of Zoology, Center for Advanced Studies, University of Rajasthan, Jaipur. The second chairperson is Dr. Priyadarshi Meena, who is Assistant Professor in Department of Zoology, University of Rajasthan, Jaipur. I humbly request the speaker to please come and dais.
सभी स्टूडेंट्स आगे आके बैठ जाएं, पीछे वाले आगे आइए, आगे बैठो।
For your kind introduction. Today I am going to talk on non-homologous end joining repair pathway of DNA double-strand breaks and its application in cancer radiotherapy. So, as we all know that DNA double-strand break repair pathways are of two types. First, homologous recombination pathway, and another one is non-homologous end joining pathway. Homologous recombination pathway accurately repairs the DNA damage using undamaged sister chromatid as a DNA template, whereas non-homologous end joining pathway is considered less accurate than HR, sometimes causing the loss of few nucleotides. As it causes the loss of few nucleotides at the junction. Homologous recombination is available only after DNA replication, that is late S and G2 phase, whereas non-homologous end joining pathway is available throughout the cell cycle and indispensable in G1 and early S phases. So this is the classical model of DNA double-strand break repair pathways. In NHEJ repair pathway, PAX, Q86, 7G, DNA PKCS, aflatoxin, PNKP, Artemis, Pol mu, Pol lambda, and XRCC4 ligase 4 complex and XLR protein works here. In the case of homologous recombination pathway, NBS1, MRE11, RAT50, RPA, BRCA2, PAL-B, and PCNA, all Pol delta and Pol epsilon is working in this repair pathway. So, NHEJ repair pathway works in the case of DNA damage. So, in the case of DNA damage, first the protein FACs and Q86 and 7G binds to the broken DNA ends. Then after that, DNA PKCS protein also binds. Then processing of overhang ends is mediated by the protein aflatoxin, PNKP, APLF, Artemis, Pol mu and Pol lambda. And after that, the XRCC4 ligase 4 complex and XLF proteins get repaired. In this pathway, this XRCC4 and ligase 4 protein and this DNA PKCS protein is very important here. And for the efficiently repair of of DNA, this kinase activity of DNA protein kinase enzyme is really very important. Is required. DNA PK inhibitor. DNA PK inhibitors such as wortmannin, Nu7026, Nu7441 sensitizes cells to radiation, whereas catalytically inactive DNA PK, otherwise intact, can restore at most partial NHG activity to DNA PK knockout cells. So there are two important missing links in this process. As I told you that DNA PK enzyme is really very important. So there are two important missing links. What is or are the bona fide substrate of DNA PK, and what is the biological significance of phosphorylation in NHG repair? So in the present study, we have mainly focused on XRCC4 protein. So what is XRCC4 protein? XRCC4 protein is an acidic protein of 39 kilodalton. Its deficient cells exhibits radiosensitivity and immunodeficiencies like DNA PKCS or Ku deficient cells, and it associated with DNA ligase 4 that finally joins the two DNA broken ends. So there are two important objectives of this present study. For in part first, we identified many phosphoryl sites in the XRCC4 of DNA PK. In part second, we just know the biological importance of lysine amino acids in order to know their nuclear localization function in the XRCC4 protein, and we also found a biological importance of extremely C-terminal region of XRCC4 in non-homologous end joining repair pathway. So in the part first, we just identified many phosphoryl sites in the XRCC4 protein. So how we know that XRCC4 is really phosphorylated by the DNA PK? So in the year 2000, our laboratory has found that the XRCC4 is phosphorylated with 100 gray X-ray radiation here. So we can be able to see a phosphorylated band here in the case of N059K cells. So our laboratory has used two kind of cell lines. One is DNA PK deficient cell line, another one is DNA PK sufficient cell line. And we have seen the phosphorylated form of XRCC4 only in the case of N059K cells. It means that this DNA PK enzyme is involved in the XRCC4 phosphorylation. Okay. In second panel, what we have used ATM deficient cells also. Okay, and here again we could be able to see a phosphorylated form of XRCC4 in the case of ATM deficient cell line also. Okay, in the case of here M059J cells that is deficient in DNA PK, we could not be able to see any kind of phosphorylation signals here, as you can see it here, here, and here. So it means that this DNA PK is involved in the phosphorylation of XRCC4 protein. So now after that, we just want to know that which site is phosphorylated in the case of XRCC4. So earlier, we know that this serine 53 is located in the case of here N-terminal region. So this serine 53 was the phosphorylation target of DNA PK. Okay, and we have seen the phosphorylation in the case of wild type, but when we have mutated this serine 53 into alanine, the phosphorylation signals were completely absent here. So it means that serine 53 is a phosphorylation target of DNA PK in the case of XRCC4 protein. Okay, so but after that we have found very exciting result. So when we use that in our in vitro experiment, so in the case of CBB staining, so we can see some bands here. So even we have mutated this serine 53 into alanine, even though we could be able to see some phosphorylation signal bands here. Okay, and again in the case of inorganic phosphate incorporation. Again, we could be able to see some phosphoryl signals here. In the case of phosphoryl specific antibody against serine 53, we could not be able to see phosphoryl signals here. It means that there may be some other phosphoryl sites besides serine 53. Okay. So in next panel, what we have used that here, we have used two kind of antibody here. So one is N-terminal epitopic antibody that raised against the N-terminal region of XRC protein here. So we could be able to see phosphoryl signals here. But in the case of C-terminal region, we could not be able to see any upper band here. Okay. So it means that our phosphoryl sites besides S53 are located in the C-terminal region here. Okay. So when we have seen the epitope of this C-terminal antibody, so we found that these serine may be a potential targets of DNA-PK in the case of XRC protein here. Okay. So in next part, we have synthesized three kind of peptides. One is wild type, that is normal. Another one is from 1 to 324 amino acids of XRCC4. And in another lane, we have used S327, 328 double mutant peptide here. Okay. And when we have used both the antibody here, and we could be able to see in the case of S327, 328 mutant, we could not be able to see phosphoryl signals here. So it means that these two serine may be a potential targets of DNA-PK in the case of XRC protein. So after that, we just mutated this as we just prepared phosphoryl specific antibody against various phosphoryl sites in the XRC protein, and we have seen in vitro phosphorylation, as we can see here in these panels. In the case of S53, A mutant, then S327 A mutant, and then S328 A mutant, we could not be able to see any phosphoryl signals here as compared to wild type. It means that these three serine were phosphorylated in the case of XRC protein by DNA-PK. This panel shows the reactivity of our antibody here. So in this case, we have used S327 antibody. So we just use decreasing titer of this antibody here again, and here in the case we have used serine 328 phosphoryl specific antibody here, and here we could not be able to see any signals here. We have not used any kind of antibody here, and this shows a control one, so we could be able to see phosphoryl signals here. Okay, so these three serine we have identified as phosphoryl target in the case of XRC protein. After that, we just want to know that whether really serine is only phosphorylated or threonine also phosphorylated in the case of XRC protein by DNA-PK. So to identify threonine phosphoryl sites, we have used very beautiful strategy here. So what we have done here, we have used 20 phosphoryl specific antibody on the basis of their possible cross reaction upon treatment with DNA-PK in the case of XRC protein. So what we have done here, we just used a bacterially expressed XRC protein and then mixed DNA-PK in the append-off, and we have seen phosphoryl signals with these two antibody, phosphothreonine 1607 antibody and also DNA-PKCS T2609 antibody here. So as we can be able to see phosphoryl signals in this case of XRC4 DNA-PK lane here, and again in this case XRC4 DNA-PK lane here with PKCS T2609 antibody and phosphothreonine 1607 antibody. Okay, so by this panel we could be able to know that threonine is also phosphorylated in the case of XRC protein. Okay, so after that we just want to know that which threonine is phosphorylated by the DNA-PK in the case of XRC protein. So what we have done here, so in the case of XRCC4, there are 18 candidate sites. Okay, so what we did that, we just focus on 6 threonine on the basis of conservedness. These 6 threonine were conserved across the species. And what we have done here, we just mutated these threonine one by one into alanine. Okay, and in the case of this 6 mutant, we could not be able to see any phosphoryl signals here. Okay, so it means that this MT6 that that is phosphorylated. Okay, in other threonine mutant, we could be able to see phosphoryl signals very well as compared to wild type. It means that this threonine is phosphorylated by the DNA PK in the case of extracellular protein. Okay, so after that, we just see the phosphoryl signals in the case of different cell lines. So here we could be able to see that these phosphoryl signals were dependent on radiation doses. So it's increased when we increase the radiation doses here. So in the case of S327, 328, and also threonine 323 here, then again by our earlier experiment, we have seen the phosphoryl signals in the case of bacterial expressed extracellular protein and also DNA PK. But in this case, we have seen these phosphoryl signals in vivo, in the cell line also here. So in first panel, we have used Votmanin as a DNA PK inhibitor. Okay, so we could be able to see significant diminution of phosphoryl signals in the case of 327, 328, and also in the case of siRNA treatment against DNA PK, and also NU7441 is a DNA PK inhibitor. We could be able to see significant inhibition of phosphoryl signals at all these 3 sites. It means that these sites were dependent on DNA PK. It means that these were phosphorylated by the DNA PK. So in second case, we have used 2 different kind of cell line. One is DNA PK proficient cell line, another is DNA PK deficient cell line, and we could be able to see a significant inhibition in the case of M015Z cells here, that is deficient of DNA PK, and again here. So and here we have used irradiation, here we have used zeosin as a DNA damaging agent here. So the phosphoryl signals at all these 3 sites were significantly inhibited. It means that DNA PK enzyme is involved in the phosphorylation of these 3 sites. In next part, we just want to know that whether these phosphorylation sites were really important in the cell or not. Okay, so what we did here, we just mutated these all phosphoryl sites into alanine. Okay, so we just mutated this threonine 323 into alanine, S327 and S328 into alanine, and we analyzed the radiosensitivity and also assess DNA damage repair capabilities of these cells. Okay, so here we could be able to see intermediate survival rate of all these 3 mutants. Here, so as you can see, in this case, this is M10 PCMV, means there is no extracellular protein here. Here, this is wild type, and all these 3 were our mutants. So it means that these all mutants shows intermediate survival rate between M10 extracellular wild type and M10 PCMV. It means that all these 3 mutants shows radiosensitivity. In next panel, we just did comet assay to assess the DNA repair capabilities of these mutants. Okay, so we have seen a very good comet tail here. So in the case of mutant 6, that is threonine 323 mutant and M10 XR64 S327 328A mutant, we could be able to see more tail movement as compared to M10 XR64. It means that when we have mutated these phosphoryl sites into alanine, they shows the defect in DNA repair capabilities here. This is very interesting experiment. So as we have seen, the phosphorylation of S327, 328 and threonine 323. Okay, in this case, we have just seen in vivo phosphorylation of S320. And this phosphorylation is very important to assess the DNA PK activity using phosphorylation specific antibody. So earlier group have identified these phosphoryl sites, but they have not observed in vivo phosphorylation of this S320. In our laboratory, we have recently reported this S320 phosphorylation sites that this happened in the cells, means in vivo phosphorylation. And this phosphorylation is very important to assess DNA repair capabilities. Okay, so here we have seen S320 phosphorylation. In the first case, here we have used HeLa cells. So this is this protein is endogenous protein here. And then after that, we have just knocked down this XRC protein by using siRNA. So we could not be able to see any phosphorylation signals here, and XRC score is also very less here. Then after that, we have just exogenously introduced XRC protein here. Okay. And we could be able to see phosphorylation here, and in the case of S320A mutant, we could not be able to see any phosphorylation signals here, and so it means that this 320 phosphorylation is mediated in the XRC protein by DNA PK. Okay. Here this shows the dose-dependent pattern of S320 phosphorylation. This phosphorylation is also increased in response to radiation dosing here. Okay, and in this case, it's very interesting thing that in earlier case we have used immunoprecipitation to collect more XRCC4 protein here. But in this case, we could be able to see this phosphorylation signals even in cell lysates also. Okay, so this phosphorylation event is very important in the cell. Here we have just seen the time response of XRC4 serine 320 phosphorylation, and its interesting thing is there we could be able to see phosphorylation even just after radiation here. Okay, so we have just collected cells just after radiation, and it persists till 240 minutes after radiation here. Okay, so here we just want to know that really DNA PK enzyme is important for this phosphorylation. So as we can see in the panel here, so we have used Nu7441 plus, okay, here. So it means that the phosphorylation signals at these sites were greatly diminished in the case of Nu7441 treatment, that is inhibitor of DNA PK. It means that this phosphorylation site is mediated by the DNA PK enzyme. Here again we have used M059G and K cells that is deficient of DNA PK enzyme or proficient of DNA PK enzyme. So we could be able to see great diminished in the case of M059G cells here. Okay, in the case of M059K cells, we have seen very good phosphorylation signals in the case of zeosyn treated cells. Okay. So, this is the summary of our DNA PK phosphorylation sites in the XRC protein. So here we have seen S327, 328, 320 and S53 phosphorylation sites. Other group also identified S260 phosphorylation sites, T233 phosphorylation sites also. But our laboratory have reported these 5 phosphorylation sites. So, and these phosphorylation sites are really very important in cancer radiotherapy, as they used to reflect the DNA PK activity, and they also used to screen out DNA PK inhibitors in the case of radiotherapy treatment. And these phosphorylation sites are also used to control the radiosensitivity, and by that we can develop new radiosensitizers. In part second, this is also very important study in our laboratory, we did it. So in first part, we just know the biological importance of lysine amino acids and also extremely C-terminal region of XRC protein. So why we used lysine? Because as we all know that the XRC protein have nuclear localization signals, and these nuclear localization signals were mediated by lysine amino acid. So this figure shows a lysine amino acid that is conserved across the many species here, and what we did that we just mutated these lysine one by one. Okay, so this is very important thing, that when we have mutated this glycine 271 into arginine, okay, so we could not be able to see any XRC protein. So as you can see, there were holes were present in the cells. It means that there is no nuclear XRC protein were present here. So it means that this lysine 271 is essential for the nuclear localization of XRC protein. So, and when we have seen the Western blotting, so this XRC protein is present in all the cell lines, even with the K271R mutant also, we could be able to see this XRC protein, but we could not be able to see XRC protein in the nucleus. Okay. And this shows a DNA repair capabilities of these mutants. So as we can see, more gamma S2X foci in the case of K271R mutant. It means that when we have mutated this K271 into arginine, it shows a defect in DNA repair capabilities here. So it means that this K27 is very important for the nuclear localization of XRC protein. So in another part, we just want to know the role of extremely C-terminal region. We have designated this as XECT domain of XRC protein. So why we have used this extremely C-terminal region? Because we have identified many phospholipid sites in this region. As we can see, there is a cluster of phospholipid sites as T323, S320, S327, and 328 sites were located in the extremely C-terminal region. So what we did here, so we have just seen the conservedness across the many species of these 20 amino acids here. And after that, we have just mutated one by one into different amino acids on the basis of their on the basis of their structure. And we have seen very interesting thing that this R2325 and N326 mutant shows a very less survival fraction in the case of radiation treated cells here. So further studied, we have just focused on N326. Okay, when we have mutated this N326 into leucine, it's also defective in nuclear localization signals. Okay, so when we have seen the structure of XRC protein, we just want to know that whether this N326 is really important for nuclearization NLS signal, but we could not be able to see any we could not be able to see any role of N326 in NLS signals. So we just want to know that why this N326 is important for nuclear localization signals. So after that, we just see the many nuclear localization signals structure. So we have seen that when we have mutated this N326 into mutated this N326 into leucine, it creates a synthetic nuclear export signal. It means that when leucine is present in place of N, it creates a synthetic nuclear export. So it exports the XRC protein from the cell. Okay, and when we have given leptomycin B, that is the inhibitor of exportin 1 and CRM1, that is nuclear export signals. Okay, so this this treatment could not able to export the XRCC4 from the cells. So here, as you can see, in the case of leptomycin treatment, we could be able to see XRC4 protein in the cell. But in earlier one, we could not be able to see XRC4 protein here. You can see holes here, And in the case of leptomycin treated cells, we could be able to see XRC4 protein in the nucleus. And when we have checked the survival fraction in the case of N326L, N326D, N326Q, and N326A mutant, it shows intermediate survival rate between M10 XRCC4 wild type and M10 PCMV that is deficient of XRC4 protein. Okay, so it means that this N326 is really important in the case of extracellular protein. Okay, so by by our all these experiments, we just want to know the role of this NHJ repair in targeting the DNA damage response in case of cancer radiotherapy. So as we all know that in the case of DNA damage, this is normal cell. There were two pathways were exist. So this normal cell was efficiently repaired this DNA damage and finally cell was survived. In the case of cancer cells, this one pathway is inactive, so the DNA repair is mediated by another pathway, pathway B. So if we know the mechanism of this NHJ repair, so we just inhibit the another pathway, and in the case of radiotherapy, when we induce DNA damage, and one pathway is already inhibited and another pathway we have inhibited by using a different kind of mimic protein, and finally cells were died. Okay, so it means that this NHJ repair pathway can be used in the case of targeting DNA damage response in cancer radiotherapy. Okay, so how we can achieve these things? So we are just synthesizing phosphorylation mimicking protein sites. Okay, so normally when we see the DNA damage response here, so this is extracellular and ligase 4 protein, and as in our study we have seen many phosphoryl sites of the extracellular protein. Okay, so when DNA damage happened, this extracellular is phosphorylated by the DNA PK, and after that some protein is interacting with these phosphoryl sites, and finally DNA gets repaired. Okay, so in this case, if we synthesize a mimicking peptide that is correspond to phosphorylation site, so this synthetic peptide sequester our protein interacting protein. Okay, and when this mimicking peptide sequester our protein that is important for DNA repair, these cells shows radio sensitivity. So by that way we can be able to we could be able to targeted DNA damage response in the case of cancer radiotherapy. And in our laboratory we are just planning to synthesize a phosphomimicking protein, and we also want to know whether this is really very important in the case of cancer radiotherapy or not. So this is a summary. Okay, I'm not going to read it. Okay, and I am acknowledging our laboratory member, and especially Dr. Ravindran Samarth also, because he did many experiments of ST20 phosphorylation antibody in the case of ST20 phosphorylation sites. And I'm also acknowledging many funding agencies. They have funded during our study. And thank you very much for your kind attention. If you have any query, any suggestion.
In radiotherapy, specifically about XCC4 protein, DNA PKs, 320, 327 and others. Thank you very much. Now I request our chairperson, Dr. Hemant Parekh, to please express his views.
Thank you very much for the exciting presentation. So the research is very significant, and we are hopeful that it will give us fruitful results. And as you have identified the sites of phosphorylation, which are key role players, and the proteins. So now the session is open for the questions. Thank you very much.
I invite Professor Madhu Kumar to felicitate our speaker.
निकल जाने को वही जगह